Hi children, I am Ravichandra. Again I came here with another topic unlike yesterday where we talked about biological classification. Today we are going to discuss about plant kingdom, a chapter which is little tricky in the sense this chapter has things to remember and also things to understand. Again, before I get into the topic, I'll tell you the purpose of this teaching or purpose of this lecture. We are at the fag end of our program. We are about to wind up this program and we are going to write this exam called NEET in few days. So going slowly is not a good idea. And the agenda is very simple. To run through this topic more faster, point number one. Point number two, to emphasize on those areas where the questions come in large numbers. Point number three, emphasis is also on remembering. And children, how to remember things when you properly categorize. So not that you are listening to me just because you are hearing something new, what you have not heard from your teacher, nothing like that. In fact, your teacher is better placed than me because he or she has more time than compared to me. I have got a limited time to discuss here. The reason why you are listening to me is I am going to put them in a systematic order and I am going to teach it more faster. Two things which you love such that you can revise this topic in a big way. Also, I am going to emphasize those areas where the questions come in large numbers, and also I am going to emphasize those areas which are not properly elaborated in your NCERT textbook. Chalo, we'll go with this chapter, children, and the topic name is Plant Kingdom. And we are going to run with this topic very, very fast. Now, Whitaker said that plant kingdom include all organisms which have got cell wall, which have autotrophic nature, which have got tissues, organs, etc. In which group, which were previously plants not in Whitaker's classification? Cyanobacteria. Now in Linnaeus classification, they were kept under plants because they have cell wall. Now they are no more a plant because Cyanobacteria are prokaryotic. They are under Monera. In Linnaeus classification, fungi was kept under plants because they contain chitinous wall. But in Whitaker's classification, fungi is kept separately in a kingdom called fungi. Diatoms and dinoflagellates, they contain cell wall. They were kept under plants along with chlorella and chlamydomonas by earlier taxonomists. But now it is very clear. Diatoms, dinoflagellates, chlorella, chlamydomonas, they are unicellular and eukaryotic. Therefore, they are kept under Protista. Chalo. So what is left out is only plants. Now we are going to discuss about a core plant kingdom. Before we get to the concept of plant kingdom, we have got types of classifications. We are going to spend at least 2 to 3 minutes here. First type of classification is artificial. What are the criteria? It is based on few morphological characters. Next, what are the merit? Scientific. It is based on science. What are the demerits? There are plenty. First demerit. It is based only on few morphological characters. Therefore, groups, the organisms which are unrelated are grouped together. Or one more way of saying, organisms which are similar are separated because the basis is only few morphological characters. Second demerit. Vegetative characters and reproductive characters are given equal importance. Why you call it as demerit? Because vegetative characters tend to get modified, children. Now, leaves can reduce to spines whenever there is less amount of water. All right, leaves are reduced to a thread-like thin filaments when the leaves are exposed to water, inside water. So you know pretty well, modifications are more faster in vegetative organs when compared to reproductive. So what are the examples of what is called artificial system of classification? One, Linnaeus. He classified plants based on number of stamens and rhizome character, only one character. Therefore, his system of classification is artificial. Even Aristotle classified plants into herbs, shrubs and trees based on the nature of stem. Therefore, even Aristotle's classification is said to be artificial. So what we are doing is, first we are talking about basis, then we are talking about merits, then we are talking about demerits, and then we are talking about the examples. Means Aristotle and Linnaeus. Finished. That's about artificial. Now we go to natural. Again, the same order. What is the basis? Based on all the morphological characters anatomical characters, information from biochemistry, embryological characters, internal characters, external characters. Majority of characters are taken as a criteria. All right, merit. Definitely, it's a natural affinities are taken into criteria, so therefore all the organisms which are similar are grouped together. Demerit, phylogeny is not taken into criteria, and I forgot to tell you one more merit. Floral characters are given more importance to vegetative characters simply because floral characters are more stable when compared to vegetative characters. Coming to the demerit, well, it is not phylogenetic. And example, don't forget this, very popular question, Bentham and Hooker classification is a wonderful example for natural system of classification. These two gentlemen classified flowering plants, not all plants, flowering plants. Your textbook clearly highlights that. Then comes the most acceptable classification, that is phylogenetic. What is the basis? Not only all the morphological characters, anatomical characters, but also phylogeny. Primitive, advanced is taken as a criteria. Organisms which are primitive are kept first, organisms which are advanced are kept last in a cladogram. Then, what is the merit? Surely, all the characters including phylogeny is taken as a criteria. What is the demerit? None. One more merit I forgot. The organisms which are of same taxa have a common ancestor, naturally. Organisms of same taxa will have a common ancestor. And the demerit, we don't see big demerits as far as this is concerned. Engler and Prandtl's classification, Hutchinson's classification are the two examples of phylogenetic. Even Whittaker's classification is phylogenetic. But the point is, we're discussing plant kingdom. So Whittaker have classified all living organisms. So therefore, I don't want to talk Whittaker classification as a criteria here because we are discussing these classifications under the chapter called plant kingdom. That's it. Now, tell me, is there any tools for phylogenetic classification? Yes. Numerical taxonomy, cytotaxonomy, chemotaxonomy can be the proper tools for phylogenetic system of classification. What is cytotaxonomy? Chromosomal shape, size, behavior taken as a criteria. Organisms which are similar in all are grouped together. All right. Second, chemotaxonomy. The chemical constituents present in plants like proteins, secondary metabolites are taken as a criteria for classifying. That is said to be chemotaxonomy. Numerical taxonomy, all the observable characters are given some numbers, codes. All right, and these codes are fed in computer, and all the organisms which have more number of overlapping of these codes are kept in one group. So all these are the tools for phylogenetic classification. I think we have done it in appropriate amount of time. So that completes the introduction part to plant kingdom. Now we start with algae. So we're going to fix some time, children, for 10 minutes here, not more than that. Algae. I'll tell you, for the last 5 years in your NEET exam, questions from algae have outnumbered from bryophytes, pteridophytes, and gymnosperms. So I want you to read this chapter with more intent because questions are coming in large numbers from this algae. With these words, we are going to straight away start with algae. Algae are major producers of ocean. 50% of carbon dioxide fixed, your textbook clearly says. 50% of carbon dioxide fixed are in fact done by algae on this earth. That's a very important point. Habitat, most of them are aquatic, all right, or very rarely they are terrestrial, but most of them are aquatic in nature. Plant body, it is thalloid, filamentous. Then chloroplasts are present with different varied chlorophyll pigments. The only group of plants which contain chlorophyll A, B, C, D, and even E are algae. No other group of plants have got this diversified chlorophyll pigments. And even the chloroplasts are of different shapes in green algae. Then after that, we talk about reserve food. It could be Floridian starch, laminarian starch. When we go to classes, we'll discuss that. And starch is stored in pyrenoids in green algae. Then we talk about the reproduction. Vegetative reproduction is by fragmentation. Asexual reproduction is mostly by zoospores. Mostly by zoospores. All brown algae, zoospores. Chlamydomonas, a green alga, zoospores. All right. Then coming to sexual reproduction, it could be isogamy, anisogamy, or oogamy. We'll discuss that in detail when we go to different classes of algae. What is the economic importance of algae? More than 70 marine species of algae are edible. Laminaria is edible. Sargassum is edible. Porphyra is edible. And some of them are used in the preparation of agar-agar, Gelidium and Gracilaria. Some of them are used as SCP, chlorella. Spirulina, of course, it's no more a plant now. It is kept under Monera. Spirulina, blue-green alga, is also an example of an SCP, single-cell protein. So this is the basic introduction to alga, children. I am not keen in elaborating this because anyhow we are going to discuss that in 3 classes of alga. So therefore, these are the introduction points. Now let us start with the first class out of 3, Chlorophaceae. Now we're going to go in a systematic order. First, habitat. They are mostly fresh water, even they can grow in salt water, brackish water, but mostly fresh water. After that, after habitat, cell wall. Outer pectin and inner cellulose. Pectin can partially dissolve in water and form a mucilaginous. Therefore, the filaments are slimy to touch. Then after that, the chlorophyll pigments. All of you know, green alga contain A and B, just like higher plants, just like euglenoids, A and B. And other pigments are also there, carotenes and xanthophylls, but A and B is more. Then reserve food, very important. Reserve food is starch and it is stored in pyrunoids. What are pyrunoids? Look here, this is the protein part of pyrunoid. The core is a protein part. Surrounding it, the starch gets added up. Surrounding this, the starch. So they are protein bodies storing starch, pyrunoids. They are present inside the chloroplast of green alga. That's about the reserve food, starch. Then we talk about the next is the asexual reproduction, vegetative reproduction, fragmentation. Asexual reproduction. It could be with the help of motile spores called zoospores, example Chlamydomonas, or non-motile spores called aplanospores, example is Spirogyra. Then coming to sexual reproduction. Sexual reproduction may be isogamy, anisogamy or oogamy. Isogamy with non-motile gametes, Spirogyra. Isogamy with motile gametes, Ulothrix. Anisogamy, Eudorina. Oogamy is seen in Chara and also in Volvox. That completes all the important points. We started with cell wall, then we talked about chlorophyll pigments, then we talked about the reserve food, then I forgot flagella. There are 2 to 8 flagella which are apical, unlike in brown they are lateral. Here there are apical flagella present. Alright, that is also a very unique character with regard to this. Now we talk about the examples. The most important is the examples of green alga. Unicellular green alga without any flagella, chlorella. Unicellular green alga with 2 flagella, Chlamydomonas. Colonial green alga with Chlamydomonas-like cells forming a colony, Volvox. Multicellular filamentous green alga with isogamy and non-motile gametes, Spirogyra. Multicellular filamentous green alga with isogamy, motile gametes with a collar, just like my collar. Collar-shaped chloroplast is Ulothrix. Anisogamy is seen in Eudorina. And oogamy is seen in Volvox and Chara. These are all the examples of green alga which I want you to remember. If you want to listen to one more time, just rewind this and listen. I don't have time to repeat one more time. These are the examples of it. So that completes green alga, children. Now we go without wasting any time, brown alga, the same order. First, habitat. Majority of them are marine. They can also seen in brackish water, but some of them are also seen in fresh water, but most of them are marine. Coming to the next one is the plant body. Now let me talk about the plant body first. The plant body may be filamentous, example ectocarpus, or contain holdfast, stipe and frond. Example for that is laminaria, dictyota, fucus. They're all examples of that. So either it is filamentous, ectocarpus, or with holdfast, stipe and frond in laminaria, porphyra and dictyota. Then after that, cell wall. Outer elgin, inner cellulose. After cell wall, pigments, chlorophyll A and C. It contains fucoxanthin, which impart brown color. Remember that. It's a xanthophyll. Then after that, reserve food. Reserve food is laminarian starch or mannitol. Laminarian starch or mannitol, reserve food. Then we talk about asexual reproduction. Asexual reproduction is with the help of zoospores. Zoospores are pear-shaped or pyriform with 2 lateral unequal flagella. Very important. Then we talk about sexual reproduction. Just like green alga, isogamy, anisogamy and oogamy is seen. And most importantly, if it is oogamy, the female sex organ is called oogonium. and inside that you see a female gamete which is larger in size, male gamete is smaller in size, and they fuse to form a diploid zygospore. These are the points which you have to remember with regard to brown alga. Don't forget, fucus shows diplonetic life cycle. Don't forget, the frond of laminaria is edible. Don't forget, ectocarpus is filamentous without holdfast type and frond. Don't forget, sargassum and laminaria, both of them the frond is edible. So these are the points. Scalps are the massive plant body of brown alga. Even iodine is obtained from them. So these are the points with brown alga which I want you to remember. Coming to red alga, same. We start with habitat. Mostly they are marine. Even though fresh water and brackish water they can be grown, but mostly they are marine. They are mostly seen in warm waters. All right. There are some which can be grown in well-lit areas, and there are some red alga which can be growing in deep waters, but mostly warmer areas. Marine, remember that. Now they're coming to the plant body. The plant body is filamentous. It can have a frond, example is Porphyra. It is filamentous and branched, looking like a higher complex plant, Polysiphonia. Very important. And don't forget, Gelidium and Gracilaria yield agar-agar, chondrus yield carrageen. 2 years back, neat question. Carrageen is obtained from chondrus. So that's also a red alga. Now, we talk about the cell wall. Cell wall contain, just now we discussed, polysulfate esters, agar, all right, carrageen. They're all the cell wall material. They're all phycocolloids. Coming to the plant body, just now we discussed about Porphyra and Polysiphonia. Porphyra contain frond, Polysiphonia highly complex plant body which is branched, look like a siphons. Chlorophyll pigments A and D. Reserve food is Floridian starch. Floridian starch is the reserve food in red alga. Remember that. That's a very important point. It resembles glycogen and amylopectin is one question which was asked 3 years back. Then we talk about the most important is the asexual reproduction. Asexual reproduction is with the help of non-motile spores called tetraspores. And sexual reproduction is very complex, and your textbook says post-fertilization changes are complex because you see carposporophyte, tetrasporophyte, a very elaborate sexual reproduction is seen. And all that is not needed. The female sex organ is called carpogonium. Inside that you see a female gamete. All that is not needed. Just remember, red, when there is a traffic, children, whenever there is a red light, what you will do? You will stop. Non-motile. You become non-motile. So remember, red alga have non-motile gametes and non-motile spores. So when you start moving, when there is a brown or yellow light, you start moving just like that. Brown alga, motile gametes and motile spores. Red alga, non-motile gametes and non-motile spores. I'm not elaborating the sexual reproduction in brown and red alga. The life cycle is haplodiplonetic in both. All right, except fucus, which is diplonetic in brown alga. All that is not a part of our syllabus. So therefore, that elaboration is not done here. That completes red alga, the most important complex forms out of all the alga. So children, this is the time which is we have allotted with regard to alga. I'm pretty sure that you understood all the points. Understood? You already done long back. What we have done is we have properly systematized this entire concept, talking about the side headings here. First is habitat, next is plant body, next is cell wall, next is chlorophyll pigments, next is flagella. I forgot to tell you, flagella are 2 to 8 in green alga, flagella are 2 lateral pyriform in both gametes and spores in brown alga, flagella are totally absent in red alga. Then we talked about asexual reproduction, vegetative. In all three it is fragmentation. Asexual reproduction is with the help of aplanospores or zoospores in green, only zoospores in brown, tetraspores, non-motile spores in red. Coming to sexual reproduction, isogamy and oogamy is very common in green and brown, whereas in red alga, majority of them shows oogamy. So with this, we wound up this topic called alga, which is looking very simple, very clear. Remember all the points what I said. Without wasting any time, we'll start the next group that is called Bryophytes. Children, from here onwards, now look here, I'll tell you, whenever I asked students which chapter in the first year part you find some difficulty in remembering, they always say that the topic plant kingdom is difficult. All right, you know why? Because there are areas not only to remember, there are areas where you have to properly understand. There are very minute areas to remember and also to understand. So therefore, what we do today is we'll make an attempt to make it more easier. And I can promise you, children, if you listen to me for another 30 to 40 minutes properly, all right, properly, and concentrate what all I'm talking now, I bet that plant kingdom will be, this is the last time you're reading, so you won't read it again. So you'll have a lot of confidence in writing the exam. So with these words, we are going to start Bryophytes. Now listen what I'm saying here. Now what I'm saying here, please concentrate here. Now, there is a small table I'm going to write on the board. Starting from here to the end up to gymnosperms, we follow this table. And you may ask me, what is the table? First, listen here. This is the table. First, remember, sporophyte. I'm starting with the word sporophyte. What is the meaning of sporophyte? Plant which can produce spore. That is called sporophyte, spore-producing plant. So naturally, I'm going to write spore here. Spore-producing plant is a sporophyte. Now, where this spore develop? Spore develop from spore mother cells. Spore develop from spore mother cells. So it's easy to remember. Yes. Where the spore mother cells are present? Inside bag. And these bags are called sporangium. And these bags are called sporangium. And where these bags are present? These bags are present on leaves, and those leaves are called sporophylls. So easy to remember. Now let us go the other way around. Sporophyte. Sporophyte contain leaves called sporophylls. Sporophylls contain sporangium. Sporangium contains spore mother cell. Spore mother cell produce spore. First, I want to divide this entire topic into two halves. First, these are the two halves. Sporophyte, sporophyll, sporangium, spore mother cell, and spore. How to remember? Plant, first it contains leaves. These leaves contain bags. Bags contain spore mother cells. They undergo reduction division, meiosis, and give rise to spore. This is one part of the table which you have to remember. One more part of the table. Now, just like sporophyte, I'm starting here with a word called gametophyte. This is interesting. Gametophyte. That is sporophyte. This is gametophyte. Now, what does gametophyte produce? You expect gametophylls, gametangium, gametic mother cells, gametes. Of course, but it may be there in some organisms. I'm not going to detail about that. Simply, I want to say that gametophyte job is to have bags, that is called male gametangium. Yes. And this male gametangium is going to produce male gametes. Correct. That's it. And this is female gametangium. Yes. And this female gametangium is going to produce female gametes. Finished. Children, this is looking simple. I want you to remember this table. This part of the table and also this part of the table. So the gametophyte produce male gametangium producing male gametes. Gametophyte produce female gametangium, female. So spore producing plant is sporophyte. Gamete producing plant is gametophyte. So simple to it. What we are going to do later is, all of you know, these gametes fuse to form zygote. And all of you know that zygote develops into embryo. Our next agenda is to join spore to gametophyte. Very simple. I draw a line here to gametophyte and join embryo to sporophyte. Very simple. Now the table looks like this. Sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, gametangium, gametes, zygote, embryo, and sporophyte. So take this here and take this there. That completes the entire. If you are able to remember this table which is there on the board, all right, then the entire plant kingdom looks the most easiest chapter out of all the kingdoms, of all the chapters what you studied. Right, now let us put this table to work for all the plants, starting with what is called, I'll start with what is called liverworts. Liverworts, that is Markensia is an example for that. Hepatocopsida. Hepatology is the study of liver. The plant body looks like the lobes of the liver, hence you call it as liverwort, right? Now, from where we should start? Shall we start with sporophyte or gametophyte? You have an option of starting from anywhere, but I want to start from here. I'll tell you why. Because in alga and bryophytes, the dominant plant body is a gametophyte. So it is very natural for us to start with gametophyte, and we go like this. We go like this. That's it. And we go like this. So isn't it? So therefore we start with gametophyte. Now listen, I'm drawing a rough diagrams, excuse me for that. All right. Now this is a gametophyte. All right. Which contain unicellular rhizoids. Unicellular rhizoids. It is green in color. It contains gemmae, which are multicellular. Gemmae, that point is important. Multicellular green. When they get separated from here, they can develop into a daughter gametophyte. They are vegetative propagules. Now, this is the male gametophyte, and this is also the female gametophyte. So it means that the gametophytes here are unisexual. They are not bisexual, they are unisexual. So first is gametophyte is over. I put a tick mark here. It is green in color, it is dominant, it contains unicellular rhizoids, it contains multicellular buds on it called gemmae, which on separation give rise to a daughter plants. Finished. What the table says. Gametophyte should produce male gametangium and female gametangium. This is a long stack. At the tip of it, you see what is called antheridium. This is the antheridium, which is said to be the male gametangium. Male gametangium is over. And that produces male gametes. It produces biflagellated spermatozoids. Biflagellated spermatozoids. That is also over. Next important point. Now let us talk about the female gametangium. So this is the gametophyte, which is female. It contains a long stack, and this is the archegonium. For the first time you see archegonium here. You don't see archegonium in alga. In alga I told you it is oogonium in brown alga, the female sex organ. Here it is called archegonium. Inside that you see an egg cell. Finished. This is gametophyte. This is male gametangium, antheridium. This is female gametangium called archegonium. This is male gametes, this is female gametes. Then what did I say? In the presence of water, this flagella can move only in the presence of water. This, they are going to fuse with the egg to form a diploid zygote. Chalo, this is the zygote now. What happens to zygote? It develops into embryo. Now, for the first time, embryo is seen in bryophytes. In alga, this zygote undergo reduction division and give rise to spores. So therefore, there is no embryo in alga. The life cycle in alga is haplonetic. Majority of green alga show haplonetic life cycle. Outside chance of that question to come, remember that. So for the first time, archegonia is seen. For the first time, embryo is seen. And also remember, bryophytes are first land plants. Because alga are aquatic, they are the first land plants, also called amphibians of plant kingdom. Now, what is an example of amphibian of plant kingdom? So, amphibian example is frog. If they ask you one more example of amphibian, say one more frog. Don't say bryophytes. The reason is very simple. Because the reason why it's called amphibian, not that it's going to grow both in water and soil just like frog. The reason why it is called amphibian of plant kingdom is it grows on soil and need water at the time of fertilization. It never grows in water. There is only one member called Rixia which grows in water. Otherwise, don't take that as an example seriously. It grows on moist soils and need water for fertilization. Therefore, they are called amphibian. So this is antheridium. So this part is over. This part is over. Zygote develops into embryo. So we have done with this part. Now what did I say? Take this embryo to sporophyte, I said. Take this line to sporophyte. Finished. Now this is going to develop into a sporophyte. Chalo. This is the sporophyte. Rough diagram I'm drawing. Sporophyte. What does the sporophyte contain? Foot, seta, and capsule. Sporophyte is over. Foot, seta, and capsule. Next, sporophyll. Phyll means leaf. Leaf is green. Now, there is nothing which is green in color. So, therefore, there is no sporophyll. Some textbook says that this capsule in some bryophytes, not here, it turns green in color. Therefore, capsule is comparable to sporophyll, but definitely there is no sporophyll. Then next is what? Sporangium. There is a bag, children. There is a bag called sporangium. All right. Sometimes these bags are there, sometimes they are not there, but let us take this for convenience. Bag. This bag is called sporangium. Inside that you see spore mother cell. They undergo reduction division and give rise to spores. Spores get liberated. What did I say? Take the spore to gametophyte. These spores on liberation, 50% of the spore develop into gametophyte with antheridia. The remaining 50% of the spores develop into gametophyte with archegonia. That completes liverwort. This is looking very simple. All right. Now, if you're finding difficulty to start from here, start from here. No, there is nothing, there is no hard and fast rule that you have to start from here. You start from here if you are finding easy. Sporophyte, foot, seta and capsule. Sporophyll, absent. Sporangium, there is a bag. Spore mother cell, yes, they are present inside it. Reduction division to produce spores, yes. Spores get liberated and reach the soil, yes. Spore develops into gametophyte. 50% of spores develop into this gametophyte. Remaining 50% develop into this gametophyte, male and female. Gametophyte produce male gametangium, female gametangium. Let me draw here, female gametangium. Then male gametes, female gametes. This fuse to form zygote. Zygote develops into embryo, embryo develops into sporophyte. Finished. If you still find some difficulty in understanding, just rewind this and read one more, listen to one more time. You'll definitely find more easier to understand. What is unique of bryophytes? The unique of bryophytes is the sporophyte is physically and physiologically dependent on gametophyte, is very unique. This sporophyte, foot, seta and capsule, is dependent on gametophyte for water, dependent on gametophyte for food in liverworts. So it's a total parasite on gametophyte is a very, very important point you have to remember. And one more unique feature of bryophytes is the sex organs, antheridia and archegonia, contain a stalk. And all of them produce only one type of spores. Just now I told you, spore mother cells. I did not say microspore or megaspore, spore mother cells. So all of them exhibit homospory without any exception. First land plants, first archegoniates, first embryophytes. These are the points which you have to remember. Apart from that, distinctly, this is a group of plants in which the sporophyte is dependent on gametophyte is a point which you have to remember. That completes this unit, children, which I'm pretty sure that all of you understood this. That completes the first group, that is called liverwort. Now, without wasting any time, now we are going to start with the next one that is said to be what is called, this time we are going to talk about the next group of plants, and this is mosses. Now we talk about mosses. Example for moss is funaria. It's called cord moss. And example, one more example for moss is polytrichum, hairy cap moss. One more example for that is sphagnum, peat moss, which is normally used in packaging material because it has got ability to hold water. Now, without wasting any time, we'll start about Bryopsida. So we have done with Hepaticopsida, liverworts. My example is Markensia. There is one member called Porella, which is leafy, which contain 2 rows of leaves on a long stem. Not a big point, but remember, liverwort. And next is Anthoceratopsida. Example is Anthoceros. We don't have any detailed discussion in our book, so therefore we are avoiding that. Coming to Bryopsida, there are 3 examples: funaria, sphagnum, polytrichum. Remember, sphagnum is peat moss. Remember, funaria. We are going to discuss that now. Polytrichum, trichome is hair, hairy cap moss. Easy way to remember. Chalo, shall we start now? Yes. Shall we go with the table? Yes. Shall we start with gametophyte? Obviously, we'll start with gametophyte. Why? Because the dominant plant body in algae and bryophytes is gametophyte. So we'll start from here. What is the first gametophyte called? This is the young gametophyte you call. This is the young gametophyte which is called protonema. Protonema. There are branches of protonema, and these branches of protonema, you call them as secondary protonema. And this secondary protonema bear many buds. And they propagate vegetatively, and they develop into what is called a leafy gametophyte. Now this is interesting. This is interesting. In liverworts, there is only one type of gametophyte which look like the lobes of the liver. All right, it is monomorphic. Whereas when it comes to mosses, the gametophyte is dimorphic. Young gametophyte is protonema, which is filamentous, explaining about the algal ancestry of bryophytes. All right, then you have seen the adult gametophyte. This is the adult gametophyte which is leafy. So young gametophyte give rise to adult gametophyte with the help of budding. So buds are going to produce adult gametophyte. This is called gametophore because it bears gametangia on it, rightly called gametophore. It contains root, stem and leaf. What did you study about analogous organs, children, in your zoology? The wings of a bird and the wings of an insect are examples of analogous because they are structurally dissimilar but functionally similar. Don't think that this word analogous is patent only for one subject. The word analogous organs can also be seen for plants. The vegetative organs of bryophytes and the vegetative organs of higher plants are wonderful examples for analogous organs. You know why? Because both of them are structurally dissimilar. No cuticle, no stomata. They are haploid. No xylem, no phloem, no cuticle, no stomata, no mesophyll. All right, they are structurally dissimilar, but functionally similar, just like higher plants. Roots, these are rhizoids, children. I forgot to tell you. Rhizoids are multicellular here, unlike in liverworts, which are unicellular. So they have roots, rhizoids, root-like structures, better word, stem-like structures called colloids, leaf-like structures called phylloids. But in your textbook, they use the word leafy. All right, nothing wrong in it. So they are structurally dissimilar, but functionally similar. Therefore, even this is an example for analogous organ, which I want you to remember. Chalo, we completed gametophyte. The interesting is it took a little time for us to talk about gametophyte because young gametophyte is protonema and the adult gametophyte is gametophore. That's a very important point to remember. Next, what? What do you see? Male gametangium, female gametangium. What did you see in liverworts like Markensia? Male and female gametangia are present on different gametophytes. Therefore, we say that gametophytes there are dioecious. But when we come here, male and female gametophytes are present on the same plant. Therefore, this gametophore is bisexual or monoecious. Now, what do you see? At the tip of the main axis, you see what is called antheridia. These are all the antheridia which are present at the tip of the main axis. Yes. Which produces biflagellated spermatozoids. And at the tip of the lateral branch, you're going to see the archegonia. At the tip of the lateral branch, you're going to see the archegonia. And this is an egg cell. Yes or no? Yes. Finished. Gametophyte. Yes, this entire thing is gametophyte. Protonema is young or juvenile. This is adult. Male gametangium, female gametangium, male gametes, female gametes. Again, just like in liverworts, in the presence of water, male gametes fuse with female gametes to form a diploid zygote. What happens next? This zygote develops into embryo, yes. And what happens next? This embryo will develop into sporophyte, which is exactly similar to liverworts. Again, you see what? Again, you see what? Foot, seta and capsule. This is the foot and this is the seta and this is the capsule. This is the sporophyte. Now no sporophyll, but capsule here is green in color, unlike in liverworts. Capsule is green in color. It is a partial parasite, not total parasite. Therefore, you can say that sporophylls are absent, but capsule is comparable to sporophyll. Then you see a bag. This bag is called sporangium. Come on, children. I don't want to waste my time. This is already discussed. And this sporangium contains spore of the cell. Spore of the cell undergo reduction division to give rise to spores. Spores get liberated. And the spores again give rise to protonema first, followed by gametophore. If you're finding difficult to start from here and go there, start from there. Sporophyte. This is sporophyte. Foot, seta and capsule. Capsule is sporophyll. Inside that there is a bag called sporangium. Inside that spore mother cell. Reduction division. Spore. Spores get liberated. Spores reach the soil. Then the spore should give rise to gametophyte. This is the young gametophyte protonema. Adult gametophyte called gametophore. Gametophytes produce male gametangium, female gametangium, male gametes, female gametes, zygote, embryo and sporophyte. That completes bryophytes. Took a little time, children, because I want you to remember this table. So that is the time-taking part of this. And I'm pretty sure that after listening to this bryophyte topic, which we discussed for 15 minutes, all right, I'm pretty sure that you will never forget this, provided if you are good in this table. So without wasting any time, we go with the next group that is called pteridophytes. Okay, pteridophytes. Now we start with pteridophytes. Very important, easy one. Now what? Pteron is equal to fighter. Fighter. Pteron is equal to, Pteron is equal to leaf. Fighter is equal to plant. All right. I'm sorry. Again, I repeat. Pteron is equal to feather. Fighter is equal to plant. So plants with feathers? No. plants with feather-like leaves are called pteridophytes. Look at ferns, which are there, ornamental plants, which you have seen many times. They have long feather-like leaves, which attracts people. So therefore, feather-like leaves. So that is the reason we call them as pteridophytes. Habitat, most of them can grow in all types of soils. Why? Because they have vasculature for the first time, vascular cryptogams. But pteridophytes, even though they are capable of growing in all types of soil, for the sake of gametophyte, it picks moist and shady areas. I'll explain that in 2 minutes. Then, the plant body, for the first time, the dominant plant body is sporophyte. So I need not start from here and go that side. So I can start directly from that side. The plant body is a sporophyte. All right, and we can go ahead with it. So without wasting any time, Psalliopsida, Lycopsida, Spinopsida, and Pteropsida are the 4 groups. Psalliopsida, Psalliotum, that is extinct. Lycopsida, Lycopodium, and Salacinella. Spinopsida, example is Equisetum. And the last is Pteropsida, example for that is Pteris, Dryopteris, Eriantum, Marsilia, Azola. They are all examples of Pteropsida. So strictly speaking, we should start with Lycopsida, example Salacinella. But I'm going to start with Pteropsida or Philicopsida. The reason is very simple, because majority of pteridophytes are ferns. And therefore, I'm going to start with that and then come back to Salacinella. How much time we are going to allow to this? Not more than 5 minutes. All right, for this ferns. Now listen carefully. 5 minutes is also a big time. Just concentrate here. Now we'll start. Now I'm not writing the table on the board because you know pretty well about the table by now. So we'll start with sporophyte. This is the sporophyte, a rhizome. Adventitious roots, no taproot. Taproots are seen for the first time in gymnosperms. Adventitious root. This is a rhizome. All right. Then these are the feather-like leaves which I'm drawing here. Feather-like leaves, pinnately compound leaves these are. These are all pinnately compound leaves. Pinnately compound leaves. Yes. This is the rachis on either side, you see it. Now, chalo. That completes the sporophyte. Adventitious roots, rhizome, and pinnately compound leaves. What do you see after sporophyte? Sporophyll. From distance, children, when you look at a fern plant, from distance, you cannot distinguish which is a vegetative leaf and which is a sporophyll. You cannot, because both of them appear green. But when you go near it, all right, on that sporophyll, you're going to see a brown colored dots on it, on the lower side. Now, this is not a vegetative leaf, children. This is the sporophyll. If you see brown colored dots on the bottom of that leaflets, then please call that leaf as a sporophyll. So this is a sporophyte. Then this is the sporophyll. This is a sporophyll. Yes. This is a sporophyte. So let me write sporophyte is A, because we are starting from here. Sporophyll is B. Then you're going to see a bag here. This bag is called sporangium. Sporangium is C. And what do you see inside the sporangium? Spore mother cell inside the sporangium. Spore mother cell. That spore mother cell will be D. Then they undergo reduction division and give rise to spore. Spore will be E. Finished. This is the sporophyte. This is the sporophyll. There is a bag called sporangium. Inside that spore mother cells, they undergo reduction division and give rise to spores. So the left hand column is over. Now you know pretty well what is the next one. Spore should give rise to what? Gametophyte. So this is the gametophyte, which is filamentous, which is thalloid, which is green in color. This is the gametophyte, which is green in color. All right. What is this gametophyte is? Look at the difference between sporophyte and gametophyte. I'll tell you the difference. Sporophyte contain adventitious roots, rhizome, and sporophylls. Gametophyte is not distinguished. It is thalloid. Sporophyte contain adventitious roots, which are multicellular. Gametophyte called prothallus. I forgot to tell you it is called prothallus. Gametophyte called prothallus contain unicellular rhizoids. Sporophyte, they have vasculature, xylem and phloem. Gametophyte doesn't have vasculature. Sporophyte have a liberty, have a license to grow in all types of soils. Gametophyte grows only in moist and shady areas because it doesn't have vasculature. Now I'll tell you the most What does the sporophyte do? The sporophyte shows a lot of sacrifice. It says, look, I can grow on all types of soils. If I start growing in desert, so I can produce sporophyll, I can produce sporangia, I can produce spore mother cell. If I throw the spores, if they reach the desert, it cannot develop into a gametophyte. Even if it develops, the gametophyte is sensitive, doesn't have vasculature, it will wither away. So the intelligent sporophyte picks moist and shady areas in spite of the fact that they can grow on all types of soils for the sake of gametophyte. Not only that, your textbook says, if I use the same terminology which is there in the textbook, the restriction of pteridophytes is seen only to narrow geographical regions for two reasons. One, the gametophyte is non-vascularized and and is very sensitive. And two, water is required for fertilization just like bryophytes here. And water is available only when the conditions are moist. Chalo. That's about the differences between sporophyte and gametophyte. Now what do you see the gametophyte? Gametophyte produce dash. This is what is called the male gametangium. I'm not drawing big diagrams here. And this is the female gametangium. Male gametangium produce multiciliated spermatozoids. Female gametangium produce egg. So what do you see here? This is a gametophyte. Yes, that is said to be prothallus. Gametophyte produce male gametangium, that is called what is called antheridium. All right, now I directly write antheridium, and this antheridium produce male gametes. Yes, and this is the gametophyte also produce archegonium, which is said to be the female gametangium. Inside that you see an egg cell. Water is needed for fertilization. Zygote is formed. Zygote develops into embryo, and this embryo will develop into a sporophyte, which is looking very fine. That's about ferns, children. Majority of ferns are homosporous. Look here, what did I say in the sporophyte? Leaf is sporophyll, sporangium. Sporangium contains spore mother cells, I said. I never said microspore or megaspore. So majority of them are what is called homosporous, is a point which you have to remember. All right, that's it. That's about ferns, which is looking very fine. All right, now it's time for us to talk about the next group, Lycopsida. This is interesting. Example for Lycopsida is Selachnella. The disadvantage of this pteridophytes topic in your textbook is they have mixed the characters of Lycopsida and ferns in one paragraph and created a lot of confusion. So what we have to do is we have to properly segregate fern from Selachnella. Then this topic should become easier. Selachnella is one troubleshooter for most of you, and I urge all of you to concentrate here what I'm discussing about Selachnella. Now, I'll draw a simple diagram for you to understand. This is the sporophyte of Selachnella. Sporophyte of Selachnella. Yes, the stem is branched. Look here. There are two types of leaves. The leaves are smaller, microphylls. Unlike in ferns, the leaves are large, called fronds or macrophylls. Here the leaves are smaller. And what did you observe? There are two types of leaves which differ in their shape and size. So that is the reason why you call them as heterophilic. Heterophyly is seen here. Remember that word. Heterophyly is a word I want you to remember. Chalo. Next is, what I'm going to do is, simple diagram I'll draw, children. I'll draw one axis down with roots at the bottom. This is called rhizophore. Phore means bearing roots. Then I want to draw one line on the top, exactly on the same plane on the top. And this is called strobilus, which contain leaves called sporophylls. So easy way to draw. This is Selachnella. This is the sporophyte. It contains roots at the base of rhizophore. Stem is prostrate, creeping on the soil. And there are two types of leaves, heterophyllae. And there is a strobilus. I'll tell you what it is. The strobilus with sporophylls on it. That completes what is called the sporophyte of Selachnella. What do you see after sporophyte? After sporophyte, the next one is sporophyll. Now listen, I have drawn the diagram there. Sporophyll. I'll draw the strobilus again. This is the strobilus. And this is a sporophyll. Now listen carefully. For the first time, we have to use the word microsporophyll and megasporophyll here. The reason is very simple. From here onwards, we are talking about examples which are heterosporous. But don't get confused. The table is same. Sporophyte After sporophyte, we have written sporophylls, isn't it? Now, in place of sporophyll, you say microsporophyll and megasporophyll. That is the only change. In place of sporangium, say microsporangium, megasporangium. In place of spore mother cell, microspore mother cell, megaspore mother cell. In place of spore, microspore, megaspore. Two types of spores, heterospory. So simple. Chalo. Now, that's the sporophyte. After sporophyte, this is the sporophyll. So what is the sporophyll? I ask you to remember, put a heading mega. So this is sporophyll with a prefix called mega, megasporophyll. Then sporophyll should have sporangium. This is the sporangium. What is the prefix I am going to use? Mega. This is called megasporangium. Now what do you see inside the sporangium? Spore mother cell. This is the spore mother cell. Alright, what happens to the spore mother cell? It undergo reduction division. Here I am drawing the spore mother cell undergo reduction division and give rise to spores. Generally there are many spore mother cells also, but here I have drawn one. It undergo reduction division and give rise to spores. What are the spores called? Surely I can call it as megaspore, megaspores. Finished. Sporophyte, sporophyll, sporangium, spore mother cell, spore. The only thing is that I put a prefix mega. Sporophyte, megasporophyll, megasporangium, megaspore mother cell, megaspores. What is the next one? You have to go to the right hand side table. Spore should give rise to gametophyte. Yes. This spore will give rise to gametophyte. Now look here. This spore, out of 3 disintegrate, this spore is going to undergo many mitotic divisions. Many mitotic divisions and develop into many cell structure. Many cell structure called gametophyte. This is going to be female gametophyte. Let me use a word, female gametophyte. That's it. Female gametophyte. And there are some archegonia on the top, and this archegonia will have an egg cell. Finished. Sporophyte, sporophyll, sporangium, spore mother cell, spore, female gametophyte, female gametangium called archegonia, inside that the egg nucleus. That's very simple to remember. Alright, the same terminology except that we put a prefix mega. Same way. What about micro? Same. At the tip of the strobilus, this is sporophyll. So I can say microsporophyll. Then after microsporophyll, in the axil, microsporangium. Inside that, microspore mother cell, reduction division, microspore. Microspore produce male gametophyte. Male gametophyte produce male gametes. So how many types of spores produce? Very popular question. 2 types of spores, microspores and megaspores, hence heterospory. 2 examples, Salaginella and Salvinia are the 2 examples which are diagonally placed in your NCERT textbook, are examples of heterospory. Salaginella and Equisetum, which are parallelly placed in NCERT textbook, are examples with strobilus. Equisetum and ferns, which are diagonally placed in textbook, are examples for homosporous. Salaginella and Salvinia, heterosporous. Equisetum and ferns, homosporous. Salaginella and Equisetum have strobilus. Very popular questions these 3 are, remember that. Mark it in your textbook, it becomes more easier. Finished. That completes this unit, children. Alright, this completes, I repeat one last time, sporophyte, megasporophyll, megasporangium, megaspore mother cell, megaspore, female gametophyte, female gametangium called archegonium, female gamete. Next, microsporophyll, microsporangium, microspore mother cell, microspore, male gametophyte, male gametangium, male gametes. Now, we are in the last minute of our class. What happens next? The male gametes which are lost from here, naturally, this is the female gametophyte, archegonium. The male gametes which are lost from here will reach what is called the female gametangium. So what happens here is this megasporangial wall degenerate. Listen carefully. This megasporangial wall, this wall degenerate. Then what happens? The male gametes fuse with the egg cell to form a diploid zygote. You may ask me, where fertilization occur? On the parent plant. Where zygote is formed? On the parent plant. Where is the embryo formed? On the parent plant. Is the parent plant providing some nurture to the developing embryo? Yes. This is just like vivipary, what you see in higher animals. So therefore, this is a precursor character for seed habit. I repeat again, very popular question. The fact that a part of embryogenesis occur on the parent sporophyte is a precursor character to seed habit. So Salagnella will not produce any seeds, but this is one of the important precursor character for seed habit. What happens next? All of you know. This entire female gametophyte with embryo will be thrown out because you don't get a daughter plant on the parent plant, isn't it? The entire female gametophyte with the embryo will be thrown out. After reaching the soil, this embryo will develop into a sporophyte. Nice. It took a little time for me to discuss about Salagnella, but I'm pretty sure that you understood the difference between heterosporous plant called Salagnella in respect to homosporous plant called ferns. This is about the Pteridophytes children. So properly segregate what is there in your textbook, the points with regard to ferns and points with regard to what is called Salagnella. That completes the important group of plants called Pteridophytes. Next, we are going to start with gymnosperms.